So, hello everybody, and I really want to say thank you for inviting me to this important meeting, and of course, letting me present my latest results on how on how our wonderful creatures respond to um, invasive alien predators like crayfish. And so, first, I will start with the outline. And I will introduce the topic. Then I will go into odonates and various invasive alien predators. Then I will set the questions, research questions, and shortly about methods, results, and then I will summarize each study separately. And um, yes, and take home message. All right. So everybody knows that humans' impact on nature is huge. We construct dams and reservoirs, and by this we just cut organism migration channels, or we drain wetlands. And of course, we also increase the rate of biological invasions, and we do that intentionally or unintentionally. And these invasive alien species can obviously change the dynamics of native species and even whole ecosystems. Also, global warming is a challenge. Steady increase in temperature changes the season length, the growth season length, and so the phenological events such as hatching or emergence date or mating events. However, it is still unclear how these multiple stressors interact with each other and how they affect our creatures. And here I have a very nice example of the invasive alien signal crayfish, which is native to North America, but is currently widely spread on the old continent. And a very nice UK studies showed that this crayfish has a very bad effect on the freshwater ecosystems. And we see on these results that are plotted on the right side that the presence of this crayfish in the environment drops the macroinvertebrate richness and abundance, especially when the crayfish population density increases. And also it drops the population, the body mass of the native bullhead, this likely through the competition for food or shelter. And the studies also showed that this crayfish might strongly affect the chlorophyll concentration, which also indicates the reduction in plant biomass, in submerged plant biomass, even at the low crayfish density. So these negative effects are caused by direct and indirect effects. So I'll talk more on the indirect effects later on. Another example is the spiny cheek crayfish, which also originates from the North America and is a very competitive species. Destroys habitats through burrowing holes in the at the bottom or in the banks. Predates on fish eggs, on native fish eggs and aquatic insects. It's able to survive extreme conditions, for example, water pollution. and it can reproduce parthenogenetically also, and this kind of increases its spread. And again, these negative effects are caused by directly or indirectly. And this species is also put on the list of the worst alien species in Europe, and this from the environmental and socio-economic, and this for the environmental and socio-economic impact. We should also remember that these American crayfish species, which are invading Europe, are also vectors of the pathogenic water fungus, crayfish plague in particular. And so these photos show the pathogenic fungus, as well as the melanized patches on the crayfish external skeleton as an effect of strong immune response against this pathogen. Okay, so now we come back to our odonates. It's been already proved that odonates respond to native predators, but this depends on the trait, and there might be a decrease or increase of the trait value in response to the predator. For example, odonates might increase or decrease the feeding rate or the activity just to reduce the encounter rate. And consequently, if they reduce the activity and feeding rate, they reduce the development rate and growth rate, and with the cost of reduced size at emergence. However, it is still not clear if and to what extent odonates recognize presence of invasive alien species in the environment. And it is not obvious if each developmental stage of the damselfly sense predator stress to the same extent. For example, damselfly eggs might respond more to crayfish cues than to fish cues, as crayfish cues predates more on the stationary prey, whereas fish, insectivorous fish, are visual predators and they hunt for moving prey, like odonate larvae. Furthermore, warming normally increases trait values, but only below the optimal temperature. And seasonality also matters, because in temperate regions, time for larval growth and development time is limited and has to be adjusted despite other stressors. And again, combined effects of these multiple stressors are difficult to predict, and there have been few studies that examined combined effects. So based on this, I set 4 research questions. And the first one reads, do predator effects differ when odonates larvae are exposed to native versus invasive alien predators? And this question goes in line with the hypothesis of naive prey hypothesis. The second question, are all developmental stages equally sensitive to predator stress? And the third one, does warming affect predator stress and their difference between predator types? So by predator types, I mean the native ones and invasive alien. And do seasonal time constraints set by different egg laying date and then hatching date affect predator stress and their difference between predator types? And for this, I used the blue-tailed damselfly, Ischnura elegans, which is actually a model species for ecological studies. and it's pretty common throughout Europe, and is not endangered and is not protected by law. It has variable volcanism, which depends primarily on the thermal conditions, and it's a generalist. Is tolerant to different stressors, including predators. So the first results were published in recently published in the journal Scientific Reports at the end of 2024. So it's quite a recent publication. So in this experiment, I collected the blue-tailed eggs from one pond, Paszowski Pond in Kraków, southern Poland. And the pond holds perch population as well as alien Danube crayfish. So more about predators use in this experiment. So perch is quite common throughout Europe, and it's a native species, obviously. It's a model predator species in this kind of studies. And then the signal crayfish is native to North America. It was introduced to Europe in the 1970s and is quite rare in Poland still, but it's spreading. It's absent in southern Poland. So in this study, the signal crayfish was used as a true alien species. Okay, so now we move to the experimental design. Yes, it looks quite complex, but well. So in this experiment, I used a non-consumptive effect, which can be easily explained by the fact that the damselfly egg and larvae are exposed to chemical cues, which are called chiromons, synthesized and released by predators, which do that unintentionally. So damselfly eggs were collected at one pond, just as I mentioned before, and then were exposed in the laboratory conditions to two different temperatures, warming and ambient. They were exposed to perch cues and invasive alien crayfish cues, or control, that is no cues, no predator cues. And at hatching, control egg group was continued as a control larval group, but eggs that were exposed to predator cues were divided into predator exposed larval group or control. So the later subgroup allowed us to check for the latent or carryover effect from egg to larval or even to adult stage, because the experiment ended at the emergence. So the traits I measured were life history traits such as egg and larval development time, wet mass at emergence, larval boldness, or larval activity, measured as number of moves within a certain time. So the temperatures I used in this experiment were the ambient one and mild warming, which were realistic temperatures, because temperatures measured in the field more or less agreed with the ambient temperature. And fish and crayfish were housed in the laboratory conditions in aquaria, and the densities of these predators were calculated based on the basal metabolic rate equations, and were more or less similar to the densities in nature. Okay, now we move to the results. Okay, so the plot, the x-axis of this plot shows the predator cues, that is control, perch, and signal crayfish cues. And the y-axis, in this case, indicates egg development time measured in days. So it's between egg laying and hatching. Okay, so late individuals had a longer egg development time, and warming reduced egg development time. But predator cues increased the value of this trait. Okay, the three-way interaction, which is very interesting, indicated that warming shortened egg development time, especially in late control individuals, which could be explained as a compensation mechanism for late egg laying. And predator cues increased development time, and most strongly in the late group exposed to signal crayfish cues in ambient temperature. And this again indicated that the compensation for late egg laying disappeared, or even went in the opposite direction. Okay, now we move to another trait. Total development time until emergence. And here, late individuals decrease development time, which could be again explained as a compensation mechanism. And warming further decreased the value of this trait, especially in late individuals, which was supported by the significant interaction between physiology and temperature. Predator cues increase development time, but showed a weaker response than eggs. So the larvae generally showed a weaker response to predator cues than eggs, which could be explained by the larval acclimation to this stressor. I also noted a carryover effect, so latent effect, but only in under signal crayfish cue treatment. The next trait measured was mass at emergence. And we see that late individuals decreased mass, but only under warming temperature, indicated by phenology by temperature. And warming increased body mass in early individuals, and which might be explained that mild warming temperature was closer to the optimal temperature than ambient temperature. However, in this trait, there was no indication, or in this case, the predator cue had no effect. So now we move to the behavioral traits. No response to predator cues in these traits, which indicates that not all traits respond to predator stress. And however, there was a temperature effect on boldness. That is, warming increased boldness, which could be explained as adaptive response. And there was also a physiological effect on number of moves. So late individuals increased their activity. So to summarize, I found a direct effect, direct native and invasive alien predator effects on egg traits, as well as direct predator effect on larval and adult traits. But these larval and adult responses were weaker than egg response. I also found a carryover effect, but only under invasive alien predator treatment. And contrary to time constraint and warming, there was no predator effect on behavioral traits, indicating that response depends on the trait measured. And the second study I will present during my talk was published, it was recently, a few days ago, published in the journal Neobiota. And in this paper, I focus on more crayfish species, on their effects on damselfly response. And yes, so we move forward. In this study, I collected odonates eggs from 2 different ponds, also within Kraków, in southern Poland. And that was Bonarka Pond and Melniki Pond. So these ponds differed had different crayfish history. So in this study, I used noble crayfish, which is native to Europe, endangered and protected by law in Poland. The second one was Danube crayfish, which is alien in Poland, not invasive, but only alien, and originates from the Caspian Sea region. Is quite uncommon in Poland. Signal crayfish, which was described in previous study. And the last one was a spiny cheek crayfish, which is also native to North America and was introduced to Europe at the end of 19th century. Is actually the most common species in Poland now, also including southern Poland. So here are maps with the crayfish distributions. Since I use as many as 4 different crayfish species, I set equally valid hypothesis, which was based on the phylogeny. And so the closer the predator species phylogenetic relatedness, the more similar the damselfly's response should be due to the similar chemical composition of crayfish chemical cue. And the experimental design. Here, egg clutches were sampled at 2 ponds, as mentioned before, and eggs were exposed to one of 4 crayfish species, cues, or a control group. At hatching, every egg clutch was divided into 2 larval subgroups. The first did not continue with crayfish cue, so these are shown by field arrows, and the second continued exposition with crayfish cues, indicated by empty arrows. And the experiment ended when larvae reached the age of 44 days since hatching. And I measured egg traits as well as larval traits, which are listed here. Okay, let's move to the results. The first plot shows egg survival across different crayfish cue treatments. And it's easy to notice that overall there was a significant crayfish cue effect and a negative effect of crayfish cue effects. And the order of significance was that signal crayfish cue decrease the survival the most, then the Danube crayfish, spiny cheek, and noble. So this suggests that the least common crayfish species in southern Poland brought the strongest response in egg survival. So the opposite to the naive prey hypothesis. No support for this hypothesis. And the pond effect was non-significant. So I will not continue talking about the phylogenetic relationship and pond effect when describing results for other traits, because in all cases these two factors were non-significant. Okay, now we focus on egg development time. So generally, eggs increased development time under crayfish cues treatment, and the strongest delay was under the signal crayfish cue, then Danube, spiny cheek, noble, and and noble crayfish cue compared to control. So the pattern was more or less similar as in egg survival. The least common crayfish species, the stronger effect. Here, strongest delay in egg development time. Okay, the third trait measured for the egg stage was in the egg stage was the hatching synchrony, and this trait was measured as coefficients of variation. So generally, crayfish Q led to more or less 2 times lower synchrony compared to control, and the synchrony did not differ between treatments with crayfish Qs. So a uniform response across different crayfish treatments. And now about larval survival until day 44. Generally decreased survival under crayfish Q. However, only under combined exposition to the noob crayfish and signal crayfish Qs. So this larval survival dropped only under signal crayfish cues and Danube crayfish cues. Interestingly, I noticed a carryover effect, but again, only under these two crayfish treatments, Danube and signal crayfish. For the mass, there was a significant crayfish Q effect, again negative, on the larval mass, which happened especially under continuous exposition to noble crayfish Q and Danube and signal crayfish Qs. The carryover effect happened only in Danube crayfish Q treatment. So this indicated the strongest effects from the least common crayfish. And for the fat content, larval fat content, generally there was a decreased fat content under crayfish Q treatment, and this happened in combined exposition to Danube, noble, and spiny cheek crayfish Qs, but not to combined egg and larval exposition to signal crayfish Q treatment. And the carryover effect happened only under signal crayfish Q treatment. And the last trait measured in this study was the immune function, measured as phenol oxidase activity. And in this trait, I found no effect of signal crayfish Q treatment. Okay, so I found the strongest egg and larval responses to the least common invasive alien and alien crayfish, and bringing no support for the naive prey hypothesis. I also found a carryover effect, but again, only under the least common crayfish species. And the damselfly responses did not follow crayfish phylogenetic relationship. So the take-home message is that global change factors may magnify the impact of invasive species on temperate odonates. And to deal with this, prevention of spread or perhaps total removal of invasive species should be considered, as was also mentioned by Professor Samways et al. In a forum paper published several days ago in the journal Insect Conservation and Diversity. Thank you for your attention. And yeah, thanks now go to collaborators, students and postdocs, as well as to financial support from National Science Center and the Institute of Nature Conservation that I work for. Thank you very much.